genomics? Well, I imagine you're all here because you're partly interested in heart disease and partly interested in genetics. So I hope I'll be able to inform you about both of these. Now, over the last few years, of course, there's been a huge upsurge in the genetic information that we have available. And you've all seen headlines like this in the press or in the evening news. And these clever scientists are finding genes for all sorts of things, sometimes for diseases like restless leg syndrome, which you've never even heard of. But what I'm interested in is when scientists find a gene for heart disease. And let me just explain to you what that catchy headline means. It means that scientists have identified a gene where a sequence change alters the level or function of the protein that gene makes. This results in a high risk of developing the disease, in this case heart disease, in the presence of an environmental stressor. Not a catchy title, so you can see why it's scientists find gene, but that really is very shorthand. So that's what I'm going to be explaining about that part. So, and then of course from the point of view of what we're trying to do in my group and others throughout the countries, how can we translate these findings to health benefit? So I'm going to talk about 4 different aspects. Firstly, what are the causes and the mechanisms of heart disease? And then I'll explain to you what is a gene and how does it work. And there's this particular concept called a SNP, and you'll go away at the end of the talk knowing exactly what a SNP is and how it can be used. And then finally, how we can use DNA tests to identify patients. And I want to acknowledge that my work is almost completely funded by the British Heart Foundation, and to acknowledge the contribution from a number of people in my group in cardiovascular genetics and from others here at UCL. So, what are the common risk factors for heart disease? So, audience participation, just shout out. What sort of things do you think about when you say risk of heart disease?
Blood pressure.
Yep. Blood pressure, good one, yep. Diet, yep. Smoking, yep. What? Diabetes, yeah. Weight, yes. Obesity, yeah. Cholesterol, yes. Yes, very good. Anything else? Family history, yes. There's the genes coming in for sure. Anything else? Gender, very good. Age, yes, I think we've got them all. Well done. And the cholesterol, I won't go into it in much detail, but in the lay press there's good cholesterol, which is HDL, and you can ask me questions about that if you want, but you obviously want lots of that. And then there's bad cholesterol, which is LDL, and obviously you want to have low levels of that. And high levels of LDL are one of the major risk factors for heart disease. So I think perhaps we've got them all except maybe stress. So the point to make about these is that some of them are modifiable and some of them are not. Clearly, you can't do anything about that. Some are genetic, these sorts of things, and some are purely environmental, but most have got a bit of both. Most of them, you can imagine that there's clearly the reason you're obese is partly because of your diet and your lack of exercise, but partly because of your genetic makeup. So, what are the processes of heart disease? And in some of the slides, I'll shorten this to CHD, which stands for coronary heart disease. So if you see that on the slide, that's what it means. So here's some specimens taken at post-mortem. This is an artery. It's opened up along its length and laid out. And you can see here a number of little, they're called fatty streaks. They're just little raised areas underneath the artery wall. And you can see that they're actually moving along in the direction of flow. So flow's going left to right. And you can see that they're also clustered around these branch points where small blood vessels go off. So clearly, flow is somehow determining this. And these are, when you look at them, they're cells, they're macrophages there, and the cells are gobbling up the bad cholesterol. And this is some of the very early part of the early development of atherosclerosis. This on the right is a much later lesion, and you can see the artery used to be this big, and now the lumen, the space for the blood, has been reduced to maybe only 30% of what it was. So clearly blood flow downstream of this is going to be reduced. And this is full of crystals of cholesterol. When you cut it, you can actually hear it sort of going crack, crack, crack. And obviously bad, you know, that's not what you want. But once it's got to this stage, there's probably not much you can do about it, whereas this stage is very easy to reverse. And this is the artery of a patient with a genetic form of very high cholesterol. I won't talk about it today. The disease is called FH, and this was a 45-year-old man who's a smoker. And instead of looking like this, the whole of this section of artery is clearly absolutely laden with cholesterol, obviously very bad news. Again, in section here, this is a piece of an artery. In life, it would have been full of blood and circular, but you can see some parts of it have been spared from the development of the lesion, and others have. The lesions often occur on one side, and here's the developing lesion. This is what it looks like when you open up. Parts of the vessel are spared, and part have got this sort of fried egg appearance. You can see cholesterol, the yellow, and then here there's some ulcers. It's starting to break down, and there's even some clot that's been formed and maybe incorporated in the plaque. That helps it to grow when this blood clot forms. And it's the blood clot that actually is the problem. Here again is an artery in section. And you can see this part of the plaque has ruptured. It's a bit like a scab coming off the skin, and the response, of course, is to form a clot to stop the bleed. When it's on your skin, that's good. When it's inside a vessel, that could be bad. You obviously want to stop it, but here what's happened is that this lesion, again, in high magnification, this clot has formed. It's full of fibrin and platelets and other things, and clearly it's stopping further blood loss. The problem occurs when a clot gets bigger and bigger and bigger and blocks up the artery, because clearly no blood will be able to get through, and it's that kills you. It's that causes the damage to the heart tissue or causes a stroke or something like that. So this is what you want to avoid. So what we know about the pathological processes of heart disease actually helps give us some clues about the genes we should be looking at. So just as an example here, over time, the arteries that we have when we're born are nice and clean and nothing going on. The fatty streaks start to develop here in our 20s or 30s, and people who are in their 50s and 60s, they get to the point where there's still enough blood flow if you're sitting watching TV, but when you run for the bus, there's not enough oxygen getting through this vessel, and the heart muscle says, oh, I'm cramping, and you get what's called angina, so chest pains on exercise. This is a very good warning system that your arteries are blocked up. Please go to see your GP to get it fixed. He'll refer you on to have tests with stents and things like this. So this is the warning sign which you ignore at your peril, because you've then got this plaque rupture, the clot forming, and then that causes the lack of oxygen and blood flow to a part of the heart, which then dies, and that's called the myocardial infarction. So that's the processes. Now, we can use this to try to think of the sorts of genes we should be looking at. Here, I've put this cascade again. We've talked about the problem of high cholesterol. So we can think of genes that are involved in carrying the cholesterol around the blood, apoproteins, receptors that remove it from the blood, enzymes that are involved in the whole process. There's some good candidate genes there. Then in this process, we can think of proteins involved in vessel wall integrity, keeping that plaque nice and safe, stopping it rupturing. Then we can think of proteins involved in the coagulation system, making the clot, breaking the clot down. But then, of course, on top of this, we've got all the environmental issues and how they interact here. And then these other diseases which you mentioned, hypertension, diabetes, obesity, some genes, some environment. So I hope you're getting the picture that it's quite complicated. This is the way that I think about it. Heart disease risk is multifactorial, and that in this example we've got, say, 11 genes that we know about in the population. And if you're lucky, you've inherited very few of the risk alleles, the alleles that are increasing your risk of heart disease. If you're unlucky, you've got lots of them. And then you can choose, to some extent, what environment you live in. You could choose an environment where you have low risk factors, or by bad luck or by bad judgment or both, you could end up in having a high-risk environment, being obese and smoking and things like that. And the model that I think is the most appropriate is, in order to develop early heart disease, you have to have a bit of both. You mainly have to have a few of these genes that are increasing your risk a bit, and a few of these environmental factors. So the research that we're trying to do has really got three aims. Firstly, to find the genes that are involved in determining risk of disease, then work out how they interact with each other and with environments such as diet and things, and then use this for patient benefit. So how can we think of using this? What's the sort of model? Well, in the UK, for various reasons that we can discuss, the guidelines are that if the doctors say, here's a man and his risk over the next 10 years of developing heart disease, having a heart attack or something like that, is more than 20%, then he should be given a statin. That's the cutoff where the government says it's cost effective. So here's a, if you like, typical UK punter. You can see he's 57. There's his LDL. He's got quite a lot of the bad, not so much of the good. He's got high triglycerides, high blood pressure. He's a smoker, family history. When the doctor uses all this information, his 10-year risk of having a heart attack is 21%. Easy. Give him a statin. That's him. I won't say it's him sorted, because clearly you want him to lose weight and get blood pressure. But from a therapeutic point of view, that's very effective. Statins work very well. They lower cholesterol. They prevent heart attacks. And now that they're relatively cheap. So from a government and NHS point of view, that's very effective. Here's his colleague. You can see he probably goes to the gym a couple of times a week. His LDL is a bit lower. His HDL is a bit higher. His triglycerides are a bit lower. Blood pressure and smoking, family history the same. That information says his 10-year risk is only 18%. So what the doctor says is, no, you should stop smoking. And he may recommend him to go to a smoking cessation clinic. But he doesn't get a statin, doesn't qualify. Now, the question is, maybe this man, partly because of his family history, has inherited 7 or 8 or 9 of these risk alleles. And so actually, if you could include that information, you might say, you really are at high risk. We really should give you a statin. So that's the model. Can we use genotype information on top of all the important risk factors we already know about to try to risk stratify, to take all the people in this group and say, right, because of your genotype, you're at high risk or not. So how do we currently estimate an individual's risk? There are a number of different algorithms. There's this one called ProCam, is based on a study in Germany. Framingham is based on a study in America. But both of them use similar sorts of approaches. They divide the risk traits into strata, into levels, and then you get points as you increase the risk. For if you have low HDL and high LDL and diabetes. So for everyone in the population, we can give them a score. And then their risk of developing heart disease goes up in some sort of curvy linear fashion like that. Okay, so that's how it works. Does it work well in practice? This is a study that we've carried out now for many years. It's based on just over 3,000 healthy middle-aged men. They were free of heart disease on entry. The study now is more than its 15th year. But in the first 10 years, there were about 200 events. And you can see here that compared to the men who remained event-free, those who subsequently had a heart attack at the baseline when we first recruited them were a bit older, were a bit fatter, had higher blood pressure, had higher cholesterol, they had higher fibrinogen, the clotting protein, more of them smoked. So very much the sort of risk profile that you'd expect. Okay, so let's put this information into the algorithm and see how many of the 200 events that occurred it actually predicts. Now, this is in a cartoon form, but I think you get the idea. This is the risk score distribution in the men who remained event-free, and in red, it's the men who went on to have an event. What you do with any test like this is you set a cutoff. You say, I'm going to say 5% false positive. So this is the score at which I will say you're at risk. Unfortunately, you're 5% of the men who don't have an event, you'll say, And what you can see is that you correctly predict 14% of the events that actually occur. What that means is you miss 86% of the events. It's a pretty rotten test. But it's being used all over this country, all over Europe, all over America, because it's the best we've got. So it doesn't do very well, but it's the best. So can we improve on this by adding genetic factors? Let's just step back a second. What we're trying to do here is predict risk of heart attack. Probably the best predictor is the amount of blockage, the stenosis, in the coronary arteries. That would be the best thing we could measure. We can't measure that cheaply or safely. It requires invasive techniques. So we use a surrogate like LDL cholesterol. You've got high LDL, you've got high coronary stenosis. So we know the proteins that are involved in determining this and the genes. And what we're trying to do, if we're lucky or clever, is find a genotype in one of these genes that will be associated with risk of MI. But what you can see is that's working through its effect on levels of LDL. So most genotypes will not predict risk over and above the measures of the things that we can do simply. The converse of that is that if we can find genes that are involved in traits not included in this algorithm, they're probably going to add best. So that's the challenge. Where can we find those genes? Okay, I'm going to spend a few minutes now talking about genes and chromosomes. I'll go through this quite quickly because I think most of you are probably aware of it. So each chromosome is the storage unit for thousands of genes. We now know we've got about 25,000 gene pairs, and the genes make us who they are. They come in 22 matching pairs, one from mum and one from dad. The genes are made up of these 4 chemical code known as DNA, A, C, G, and T, and here's the iconic Watson and Crick double helix. Each gene can be many thousands of bases long, and the information is a code that contains the information to make a whole human being. When a change occurs in the DNA sequence, the genetic instructions are no longer correct. We can't see these, but we can work out how they are using biochemical techniques. And it's like a typographical error has been made. So the manual on page 75, instead of saying cat at one place, it says tat. And then the cell makes a part that doesn't work. And that's the problem. So just to give you an illustration, imagine the manual tells how to make an umbrella. So here's the gene, and the gene, when a certain environment comes along, it makes a protein which protects the organism from that environment. So there's the gene working nicely. So here's the gene, and it's got a mutation. And it can have an effect in several different ways. It could be that it just doesn't make enough of the protective protein. Or it could be that it makes a protective protein that just doesn't function very well. Just when you want it, the thing closes. Or it could be that it somehow affects the timing of when the protein's made. So instead of making it in the right time, it makes it in the wrong time. So the thing to remember is that genes make proteins that are important for health, and mutations stop them working. There are a number of different types of genetic disease: whole chromosomes, Down syndrome as an example, single gene defects, and multifactorial. And of course, we're talking about multifactorial. Again, the thing to remember is that the clinical consequence of any disease is always modified by environment. Some diseases are strongly genetic and some are strongly influenced by environment. You would all guess that hair colour is essentially a genetic trait, but we can modify it, and probably several in the audience are busy modifying it, out of a bottle. And of course, height is very strongly genetic, but your nutrition as a child has a very strong influence on whether you attain your genetic potential or not. So here's an example of a chromosomal disorder, Down syndrome, 3 copies of chromosome 21. The Mendelian forms, the ones that we're most interested in, this is a picture of our hero, Gregor Mendel. He was a monk in Czechoslovakia. He developed the laws of genetics based on studying peas. And just to give you just a very brief feel of this, there are 2 important differences about tell you what, I'm running out of time, so I'm going to skip that one. The thing to remember is that chance has no memory. So each individual in the family has an equal chance of getting the bad gene, whether or not someone else in the family has already got it. So with heart disease, I'm sure I've persuaded you now that really we're talking about a multifactorial situation. So although we might find some individuals in the population, they've got a mutation that has a very large impact on an individual's risk. This disorder that I briefly mentioned, FH, is an example. Most of the people, we're talking about mutations of small impact. That histogram with with the 11 different genes. I've given you the names of some of them. It's polygenic, and we've got this environment. So this, if you like, is the challenge. We've got to work out how, we're not talking about a single gene disorder, we're talking about putting together information from lots of genes. Okay, so what's a SNP? SNP stands for single nucleotide polymorphism. What this part means is that there's one base in the DNA that's changed. Polymorphism is from the Greek, it means many shapes. So a SNP says there's a difference in a DNA sequence that's found commonly in the population, and by definition we use over 1%. So here it is, there's a sequence on one chromosome that I got from mum, and the sequence that I got from dad has got that T changed to an A. And these might be mutations, they might have functions, as I showed you, but the majority of SNPs occur outside the gene and are of unknown or probably of no effect at all. That doesn't matter, we can still use them as genetic tools. They've proved to be very, very useful. And all SNPs have got a unique ID number, a reference sequence number, which now we've got millions of them, so you've got 7 or 8 figure numbers. Hard to keep them straight. What we've been able to do over the last 20 years, 30 years, and this has really changed the way we can do things, is the whole process of determining genotype has got faster and faster. In the 70s, we could do maybe 20 genotypes a week. In the 80s, we could do maybe 200 or 2,000. In the 90s, we were up to more than 5,000. Now we're in the warp drive area, where we can really generate vast amounts of data every day. And of course, what we need to handle all these is databases, and these are publicly available, supported by the Wellcome Trust, the MRC, the NIH, to handle all this information, to handle the bioinformatics. We can now capture data from the whole genome in one experiment. And this is the way it's done. So you might take 1,000 cases here with heart disease and 1,000 controls. You would then use this commercially available chip and genotype each of these individuals for maybe 300,000 or maybe 1 million SNPs. And you can really do this very quickly. And I've shown them here. They cover the entire genome. What you're doing here is looking for a frequency difference between the cases and the controls. Is this particular SNP more frequent in the cases? It would indicate it's predisposing to heart disease than in the controls. The problem, of course, if you're doing 1 million different contrasts is you're going to get a lot of differences that will be statistically significant by chance alone. So you have to have your threshold for significance very low. So your p-value has got to be very low. But you may still include some of those by chance, and what you now do is you replicate in a second study, but you only use those maybe 20 or 30 SNPs that were significant in the first one, so you just genotype in the second sample for those SNPs. And if something is still statistically significant, you say, right, we have found the gene for restless leg syndrome or whatever it is you're interested in. The data from these genome-wide association studies is presented as something that's called a Manhattan plot. And it's presented as the p-value, the statistical significance, per SNP per chromosome. And what everyone was hoping was that there would be lots of statistically significant hits, so it would look like the Manhattan skyline. When the paper was, 3 papers were published in 2007 looking at heart disease, this is what it looked like. There was essentially only one SNP on chromosome 9, and so it's rather more like the Oxford skyline than the Manhattan skyline. However, this very important finding has been replicated in many independent data sets, and it really is a major breakthrough. This particular SNP is 58,000 bases from the nearest gene, which are called CDKN2A and 2B. They're genes involved in cell cycling, so telling the cell whether to replicate or not. No one would have thought in their wildest dreams that they were a candidate gene for heart disease. The common SNPs there are strongly associated with risk. That's enough p-value, enough noughts for anybody. And compared to people who've got 2 copies of the A allele, if you've got the AG, you have a 30% higher risk, or the GG, a 60% higher risk. There is no association with any heart disease traits. These GG people don't have high cholesterol or blood pressure or anything like that. And the mechanism is still unclear. So will this be useful in doing what we want to do, to identify people who are at risk? Well, the experts certainly think it will. The scientific writer from the Science magazine said, yep, they're going to lead to a genetic test. The Guardian was a little more sober. DNA find may identify. Francis Collins, who is the director of the American National Human Genome Research Institute, says, I think this is a stunner. And Carrie Stephenson, who's the CEO of Decode, the company who was one of the first people published it, said, yep, he's going to make this into a commercial test. And he has. You can buy this test from 2 different companies. This is 23andMe. For $399, you can send in a sample, and they will tell you your risk of all these and lots of other diseases based on this, for heart disease, based on this single SNP. There is a health warning. The results presented have not been cleared or approved by the FDA. Decode does the same thing, and they just use this one SNP, and here's this 1.6 and 1.3 that I mentioned. So, okay, how does it work in UK individuals? So we went back to the Northwick Park study that I told you about, and we genotyped them all. And we set the hazard ratio, the risk of having heart disease over 10 years in the AA group to be 1. The AG group, it was 1.38, and the GG group, it was 1.57. Very much in parallel with what was published. This is a Kaplan-Meier survival plot. So all individuals survived at baseline, and then you can see this is in blue, the AA group. This is the rate at which people are having heart attacks or dying, and in the AG and the GG group, it's much faster. So this really looks good. Question is, will it add significantly to our ability to predict risk over and above what we already can do, the classical risk factors? The metrics that's used is not a very intuitive one, and I won't spend any time explaining it in detail. It's called the ROC value, and the thing to remember is that if the ROC value is 0.5, it's chance, and if the ROC value is 1, it's a perfect test. This is the ROC value for the Framingham alone. It's 0.62, so it's better than 0.5, but nowhere near 1. And you can see the curve in blue here. When we do this and now add the chromosome 9 SNP, it goes up from 0.62 to 0.64. You can see the red line is closer to 1, but it hasn't moved very much. It's a 3% improvement, and 0.64 is not significantly higher than 0.62. So just as any single classical risk factor doesn't give you good prediction, no single genotype is going to have clinical utility. Save your money. What we need to do, clearly, I hope everyone's got the message by now, we've got to have several SNPs together. And we did some modelling, and we figured out that if we had 3 similar SNPs, similar sort of characteristics as the chromosome 9 one, the ROC value would go up to 0.7, and that really would be statistically significant. We didn't have to wait long to find these other SNPs. And over the last 2 or 3 years, a whole slew of genome-wide studies, you can see them all here, have found another whole bunch of genes. You can see that the chromosome 9 one is still leading the field. It's still got the highest value. The effect size for most of these is less. It's rather modest. But the risk frequency of the alleles is really quite high. We still don't know what many of these genes do. We don't know if they're associated with traits. We don't know what the functional SNPs, the real mutations, are in these genes. And we think that they will, but we're still getting the data to demonstrate that they will have clinical utility when added to the risk algorithm. So that's the piece of research that we're doing now. So what are the criteria that we need to set to have a useful heart disease risk DNA test? Clearly, it's got to have predictive power over and above the other risk factors. I'm quite certain that when we have 5 or 6 or 7 or 8 or 9 or 10 or 12 of these, it will. I don't have any problem with that. It's got to take the interaction between the genes and interaction between genes and environment into account. And currently, they don't. The model we use is simply additive. And that's probably a poor model. So we've got some work to do there. Clearly, it's got to be based on accurate and reproducible risk estimates. And we can generate the data now very quickly, so we don't have a trouble with that. Not be associated with negative psychological impact. And just in the last couple of minutes, I'll explain about that a bit. So what's the concern about the psychological impact? Well, this is the ethical, legal, and social aspects of DNA information. And here are two ladies. They've just come from the clinic. They've been given their genetic risk. They've gone home. They've lit up fags. They're knocking back the wine. And one of them, Frida says, I haven't got the gene, so it's fine for me to smoke. Fanny says, I've got the gene, so I guess I'm doomed anyway. So this is the concern. False reassurance and fatalism. And it does appear now that we've got more information about how this works, is that these concerns are unjustified. The studies that we've carried out and others suggest that DNA risk information doesn't go to greater fatalism if you present it in the right way. And it may actually motivate people to change behavior. They say, I've got a genetic risk. I really must take my statins. I really must quit smoking. It really helps to motivate them. So I think we don't have concerns about that. So finally, I'm quite convinced, to go back to the title I gave, fact or fiction, I think that DNA tests will be possible. I think they are a fact. We've clearly got to use several genes. We've got to base them on accurate and reproducible risk estimates. And currently, we only have data in white, middle-aged men. I'm sorry, ladies, I don't know whether this algorithm will work in ladies, in people from different ethnic groups. We don't have enough data on that aspect. So that's clearly an area we've got to work. We've got to do a lot of work. You must use them with the risk factors. The genetics themselves don't help, which is why, in my view, the internet tests are not valuable, and I think they're probably actually misleading and not helpful. I really do think they're going to help to risk stratify in, for example, the CHD risk clinics that are carried out in UCLH. And the way we would imagine doing it is when someone is referred by their GP to a risk clinic, the referral letter comes back to them and says, please take a mouth swab and send it to us, and your appointment is in 2 weeks' time. By then, we've done the gene test, and when they come, we present to them data like this. We say, OK, this is your 10-year risk of heart disease based on your classical risk factors, your lipids and the rest of it. And you can see, well, it's not 20%, so that's good. However, when we add your genetic risk information, it's over 20%, and so we should give you a statin. This would be someone of average age and of your age and gender who's got average risk factors. So you can see your risk is much higher. Now will you take the statin? So you work with them to explain this. And I think then we really can prevent heart attacks. So finally, I do think we've got to work out how to give them this information, because it's understanding. If they can understand it, they're going to be able to use it. So yes, heart disease testing is ready now. And I'll stop there.
Thank you very much, Professor Humphries, for almost illuminating this slightly scary talk. I speak as a white privileged man. It is the 50s, just for good measure. So if there are any questions, I'm sure there will be. Please put up your hand, and or we'll give you a microphone.
In 1973, Professor Söderström from Malmö University gave a talk at the Royal College of Physicians entitled "What Do We Tell the Politicians About Heart Disease?" 37 years later, what do we tell them?
Yes. Yes. It's a real challenge. One of the pieces of work that I do is on this monogenic form of early heart disease, where people have inherited a single gene that causes their cholesterol to be very high. Half the men with this disease will have had a heart attack by the age of 55, half the women by the age of 60 or 65. So it's a single gene disorder, and we can treat it with statins. We've demonstrated with others how well people respond. I worked with NICE, the NICE guidelines, the government organisation. 2 years ago, we published a report based on the evidence that says all these patients should be given a DNA test, use the DNA test to test their relatives. Deaf ears. There is not one service in this country that's been commissioned to do this based on the NICE guidelines. And of course, the problem is money. And I have to say that in the current financial climate, whoever wins the next election is going to be cutting the services that are carried out in the NHS. So people like me have to make this sort of thing very, very much up front. Unfortunately, it's competition, and I do appreciate that. It's a question of what the NHS wants to do is get people out of beds, firstly stop them getting into the beds, and then get them out quickly. And this sort of thing prevents events 5 or 10 years down the line when the current managers and the current politicians will be off doing something else. So it's really hard argument. And I'm afraid we need educated people like you and patient groups to try to push the politicians. It would also be very useful to have politicians who've got these sorts of diseases. That gets them really interested. I'm making a joke of it, but I think you can all appreciate that, how useful that is.
Are there studies looking at the association between SNPs and cardiovascular risk factors?
Yes, there are. It was one of the slides I skipped. It's an area that we're very interested in because we're trying to understand what we call the genetic architecture of, for example, LDL cholesterol. What are the genes and proteins that are involved? Because what we can do is, once we know that a particular protein or enzyme is involved in determining high levels of cholesterol, we could imagine a company, a pharmaceutical company, making a drug to hit that enzyme. In fact, that is the way it works. The statins block a particular enzyme in the liver, and that means that more cholesterol is taken out of the circulation. So the paradigm works very well. So understanding not just the genes we know, but finding new genes that we don't know about and we've never known about that are determining cholesterol levels or blood pressure or diabetes, insulin and things like that, give us whole insight into new therapies. And that's really a really very exciting area where this research is. That's the other side of the coin. I've just focused on the identifying patients, but developing new therapies is the other benefit. Come on, this side of the audience has got to think of some questions now. Ursula's got a question. No. Yes.
Given that the SNP was sort of way away from the coding region of DNA, do you know or can you speculate why on earth it is associated with increased risk?
Yes, large amounts of speculation and large amounts of grant money going in to try to figure it out. It's fair to say we don't know. One of the things, but we can wave our hands a lot. One of the things that happens that we've realized recently when we have all these SNPs is that large chunks of the DNA, the chromosomes, are inherited together. So if you like, SNPs are correlated. They're related to each other. So this SNP might be it might be correlated with a SNP in the gene of interest. Might not be these genes. It might actually be correlated with a marker down there. The other thing that we know recently, we've again just been able to really measure, is that control of expression of the genes is sometimes worked out by making a small piece of RNA, which then affects the half-life of the RNA of interest. And it's a whole new area that people really didn't even think about 5 or 10 years ago. So it may be that this SNP is affecting the production or the shape of an RNA that's somehow affecting maybe not these genes, maybe some other genes downstream at all. So there's a number of different mechanisms, but we don't know at the moment.
Thank you very much. I think we probably...
Ursula does have one question. Quick, this side of the audience, very quick.
This is about the first so-called stunning SNP.
Yeah.
You said the mechanism, how it works, isn't known yet.
Yeah.
But on the slide, there was clearly apoptosis.
Yes.
Is this possibility being investigated?
Yes, it is. Yes. We're doing some work in our group, and I can explain to you about that. But the idea would be that, just for people who don't know, the apoptosis process is where a cell goes into programmed death. So instead of just dying and making a mess, it very cunningly wraps itself up and then sort of vanishes. And it's a very safe way for cells to die. Now, what I've told you is that plaque rupture is the very key part of what's causing heart attacks. So if you could prevent plaque rupture by keeping cells alive, you might prevent plaque rupture. So the mechanism might be if this